Okay, so in this fifth and last segment, we're going to finish talking about what's going on in the small intestine and then move into the large intestine. So as I said in the previous video, the small intestine has three major parts. There's the duodenum, the jejunum, and the ileum. In the duodenum, there's a lot going on. Remember, we had uh, carbohydrate digestion, protein digestion continuing with the introduction of the pancreatic enzymes. And then we had the fat digestion that begins and also ends in the duodenum. So the duodenum, a lot is going on in the duodenum. Carbohydrate digestion and protein digestion resumes, I should say, in the duodenum, but it also continues in the jejunum and the ileum. And so by the time we leave the small intestine, everything digestible, every kind of food that digestible has been digested and absorbed. And so everything that moves on into the large intestine should be undigestible fiber or material and waste. Now, in terms of motility in the small intestine, there's only one form of motility, and that is segmentation contraction. And segmentation contractions are best suited as a mixing movement, but in the small intestine, it's actually a propulsive movement as well. It's a very slow propulsive movement, but that's okay. We don't really need propulsion through the small intestine to be fast. We actually would like it to be slow to give ample opportunity for digestion and absorption. But the reason why why segmentation contraction can be a propulsive movement is because the frequency of contractions in the duodenum and then moving down, the frequency of the contractions is going down as we move down the small intestine. So the frequency of contractions in the duodenum starts at around 12 contractions per minute, and then as we move down towards the ileum, it gets more slow. It gets to be 9 contractions per minute. And so we end up with this slowly propulsive movement, because we are contracting faster in the upper small intestine than in the lower small intestine. Okay? And so the intensity of the smooth muscle contractions involved in segmentation really depends on the degree of distension there is, so how much volume of material is in the small intestine, how much gastrin there is. So we know at the beginning of consuming a meal, we get this really big wave of gastrin. And also extrinsic nerve activity, so the vagus nerve, right? So is the brain, is the vagus nerve stimulating the movement of food through the stomach and the small intestine? So all of these things really affects the intensity of the contractions in the small intestine. But for segmentation contraction, it is achieving both a propulsion and a mixing. We also see that the small intestine, the major thing that's happening is digestion and absorption. And everything about the small intestine is really adapted for maximal digestion and absorption. So if we look at the anatomy, especially the microanatomy of that mucosal lining, what we see are many adaptations to maximize digestion and absorption. In terms of digestion, we have these membrane-bound enzymes. We don't need to secrete these enzymes because they are always available. They're actually part of the plasma membrane on the mucosal cells in the small intestine. So these membrane-bound enzymes are sometimes called brush border enzymes because the mucosal boundary, the intermucosal lining of the small intestine, has these microvilli that make it look like bristles of brush. But there are 3 types of brush border or membrane-bound enzymes. We have enterokinase, and remember the enterokinases are there to activate trypsin. Okay. So enterokinase, it was there to activate trypsin. Trypsin was secreted by the pancreas in the form of trypsinogen, and then trypsin goes and activates carboxypeptidase and chymotrypsin. Also bound to the brush border membrane is the disaccharidases, so sucrase, maltase, lactase. And then we also have aminopeptidase, and aminopeptidase was the last enzyme to break down proteins. Aminopeptidase broke down those small polypeptide fragments after all of those other proteolytic enzymes broke up the larger protein. So if we look at so obviously the brush border membrane, the membrane of the mucosal lining of the small intestine, is highly adapted for digestion. It literally has digestive enzymes embedded in the membrane. But also it's highly adapted for absorption. And the one of the aspects that helps it be adapted for absorption is the expansion and maximization of the surface area available for absorption. So we talked about how the small intestine is the length of a tennis court, packaged into a small space in the abdominal cavity. So that definitely helps hide a lot of surface area available for absorption. And then when we look at the mucosal lining, we see that the mucosal lining is organized into these tall villi. And that those villi also expand the surface area available for absorption. But then that's not it, right? Not only do we have these tall villi, if we look at each individual mucosal cell or epithelial cell that lines the villi, what we find is that on the luminal membrane, we have little microvilli, right? And those microvilli help expand even further the surface area available for absorption. And that microvilli is why the membrane is called the brush border membrane, because those microvilli look like bristles on a brush, right? And it has those embedded enzymes, those brush border enzymes, the enterokinases, the aminopeptidases, the disaccharidases. And if we look at all of this layering of villi and microvilli and coiled small intestine, what we find is that this increases the surface area available for digestion and absorption by a factor of 600, so 600 times. And that's compared to just having a flat surface be the mucosal lining of the small intestine. So it dramatically increases the surface area available for both digestion and absorption. On top of that, on top of the surface area issue, when we look at the inside of one of these villi, what we find are a tiny little network of capillaries that are there just ready to absorb anything that crosses that brush border membrane. So each little villi has its own little capillary network just ready and waiting to receive and absorb all the stuff that's coming across the brush border membrane. And then woven at the center of this capillary bed is a lymphatic vessel called the central lacteal. Now this central lacteal, like I said, it's a lymphatic vessel, and this is where fat that gets absorbed initially enters. So fat that gets absorbed along the brush border membrane does not immediately enter the bloodstream. It first enters these little lymphatic vessels called the central lacteal. And then the lymphatic flow, all lymphatic flow eventually enters the bloodstream closer to the vena cava of the heart. So all of that fat that gets absorbed eventually does make it back into the bloodstream, but initially it's not absorbed into the blood. Initially it's absorbed into the lymphatic system. And that central lacteal is ready to receive that fat that's absorbed. So let's take a look for a second at the mechanism by which this food is going to cross the mucosal lining of the small intestine, where its final digestive steps are, and where it gets absorbed. So here we've got a little cartoon representation of one of these brush border cells. And of course, this top part of the image is the brush border that's facing the lumen of the small intestine. And then underneath is this underlying capillary bed, right? That's there to receive whatever's being absorbed. So this image is representing the digestion and absorption of carbohydrates. So by the time we get to the small intestine, what we're left with are just the disaccharides, right? Because those big polysaccharides were broken up by amylase, either in the mouth by salivary amylase or in the duodenum and the small intestine by pancreatic amylase. But we're still left with those disaccharides. We've got maltose, sucrose, and lactose. So attached to the brush border membrane is maltase, that's that capital M, sucrase, that capital S, and lactase, that capital L. Maltase breaks up maltose into two glucoses. Sucrase breaks up sucrose into a glucose and a fructose. And lactase breaks up lactose into a glucose and a galactose. Once we get to those monosaccharides, each of those monosaccharides are absorbed in a slightly different way. So glucose gets absorbed with a co-transporter with sodium. Okay, so glucose is actively carried across the brush border membrane using that secondary active transporter that utilizes the sodium concentration gradient. Fructose is able to enter with using just a passive channel, and galactose enters similar to glucose with a co-transporter that also uses the sodium concentration gradient. Once inside, all of those monosaccharides, glucose, fructose, and galactose, are going to passively cross the other membrane, right? The membrane that opposes the capillaries. And as they cross that membrane, they're able to easily just go straight into the bloodstream there, into the capillaries. Now let's look at this same process for proteins. So again, by the time we get to the small intestine, those large, massive proteins have already been broken up by all of those proteolytic enzymes. It's been broken up by pepsin in the stomach. It's been broken up by trypsin, chymotrypsin, carboxypeptidase that was secreted by the pancreas. And so now, instead of a giant protein, we just have these little polypeptide fragments, right? And those little polypeptide fragments are going to be broken up and digested finally by the membrane-bound aminopeptidase in the small intestine. And then that is going to liberate the individual amino acids. Remember, there's 20 different amino acids. So along the brush border membrane, we do have an amino acid sodium cotransporter. It uses secondary active transport in the form of that sodium concentration gradient to actively move amino acids across that membrane. Once inside, amino acids cross passively out of the cell into the underlying capillaries. Now, when it comes to the digestion and absorption of fat, it's a little more complex, right? It's a little more complex because trying to digest fat in the midst of an aqueous solution is going to be challenging because fat does not dissolve in an aqueous solution. And so what we have to do is actually constantly rearrange and handle that fat in a way that makes it possible to happen in an aqueous solution. So the first step, the first two steps actually, we already kind of talked about. The first step was the emulsification of fat. And that was where we had those large droplets of fat that through intestinal mixing, we broke them up into smaller droplets of fat. Then those small droplets of fat were mixed with bile salts so that they wouldn't reform, right? And so we had these lipid emulsion droplets. Then those lipid emulsion droplets were incorporated with pancreatic lipase. The lipase broke down the triglycerides into monosaccharides and free fatty acids. Those monosaccharides and free fatty acids recombined with the bile salts to form those micelles. Okay. And then now those micelles, they are actually able to cross that brush border membrane. Okay. The micelles are easily able to cross those brush border membranes. And so this is where it becomes a little strange, because once inside that mucosal cell of the small intestine, the digested fat actually reforms triglycerides. And those triglycerides get repackaged by those cells of the small intestine into these lipoproteins that are called chylomicrons. And we've talked about those lipoproteins before, but those lipoproteins called chylomicrons, they are the packages of triglycerides that allow fat to travel in the bloodstream. So those chylomicrons initially get absorbed into that central lacteal and then eventually make it into the bloodstream. Now, some fatty acids within the mucosal cell can actually cross the other side and get directly into underlying capillaries, but mostly the monoglycerides and the fatty acids are reforming triglycerides, getting repackaged into chylomicrons, and then those chylomicrons enter the central lacteal. Okay. And those chylomicrons, those lipoproteins, they are the way that the that triglycerides get delivered throughout the body. And we studied this when we learned about hyperlipidemia, right, in Patho 1. When we studied the cardiovascular system and we learned about high cholesterol, we learned about the different lipoproteins, right, chylomicrons, HDLs, LDLs. And so this is where those chylomicrons come from. They are formed by the mucosal cells in the digestive tract, in the small intestine. Okay, so now, right, we've taken this digestive journey from the mouth to the esophagus to the stomach to the small intestine, and so now we're ready to take it to the final organ, which is the large intestine. So in the large intestine, by the time this material gets to the large intestine, all food that is digestible has been digested, all food that's absorbable has been absorbed. And so what enters the large intestine is undigestible material, at least by us, and just that represents basically waste. So in the large intestine, there is motility. We'll talk about this, and actually the large intestine has both waves of peristalsis and segmentation contractions. We do have secretion. There's a lot of mucus secretion in the large intestine. We don't have any enzymes, digestive enzymes, because we're not digesting anything. We don't have any digestion. We do have absorption, however. We do have absorption, but it's not of food. In the large intestine, we have a significant amount of water that gets reabsorbed, a significant amount of electrolytes that get reabsorbed, and also minerals, calcium, iron. Now, most of the water, electrolytes, and minerals that we absorb is absorbed in the small intestine, but the absorption that happens in the large intestine is important and significant. If we didn't have that absorption in the large intestine, we would dehydrate quite quickly, right? And we would fall into both electrolyte imbalance and mineral deficiencies quite quickly. So what's happening in the large intestine is more than just a story about the movement of waste. It's still very much important in the regulation of our fluid and electrolyte balance and mineral balance. So the first part of the large intestine starts at the cecum. At the cecum is where the small intestine empties into the large intestine. And the opening to the large intestine from the small intestine is called the ileocecal valve. It's right there at that cecum that we have the structure that's described as being vestigial or left over from evolution, which is the appendix. Okay. And so then from the cecum, we move into what's called the ascending colon. Okay, that ascending colon. From the ascending colon, we move into the transverse colon, right? That moves from the right side of the person to the left side. From there, we get to the descending colon, and then this sort of curved S-shaped sigmoid colon, and then finally to the rectum. Okay. And then at the bottom of that rectum, we have our anal sphincter. And the anal sphincter, there's actually two parts to the anal sphincter. We have the internal anal sphincter, which is composed of smooth muscle and it's controlled involuntarily. And then we have the external anal sphincter, which is composed of skeletal muscle, which is controlled voluntarily. Okay. So those are the basic components of the large intestine. You notice that there is this line on both sides of the large intestine called the tinea coli. This is just a connective tissue that helps provide structure to the large intestine. And also notice that the large intestine is sort of separated or forms these sort of rings called haustra. Okay. So when we think about motility in the large intestine, we do have a form of motility that is peristalsis, and we also have a form of segmentation contraction. The segmentation contraction that happens in the large intestine is called Haustral contractions. These are slow, they're non-propulsive, they're really the result of this autonomous, rhythmic, colonic, smooth muscle contraction. They happen at around 2 to 3 an hour, so they're really slow. They do represent a mixing movement, and their purpose is really to increase the contact between the contents of the large intestine and the mucosal lining. And that really helps enhance absorption of water, electrolytes, minerals. And these haustral contractions are controlled by local reflexes, mostly by the enteric nervous system, specifically the myenteric plexus. The other form of motility are these waves of peristalsis. In the large intestine, we call these waves of peristalsis mass movements. It is a very strong propulsive movement. They tend to occur about 3 to 4 times per day, and they tend to get triggered by gastrin. They get triggered when we consume food. Remember, gastrin was a hormone produced by those G cells in the stomach, and what releases gastrin is the anticipatory cues involved in eating. So thinking about food, getting hungry, smelling food, tasting food, swallowing food, chewing food. And also the presence of food in the stomach also releases gastrin. So eating and digesting food, having food enter our bodies and enter our stomach, releases gastrin, and gastrin is what's going to trigger these mass movements. And these mass movements, they're very strongly propulsive. They're able to drive feces either the entire length of the colon or 3/4 of the length of the colon within a few seconds. Okay? And if you have enough bulk in your colon and you get these mass movements, they may trigger a bowel movement. It may trigger defecation. And so you might find that if you are somebody who gets a decent amount of fiber, you have a healthy colon, you might end up having a bowel movement 3 times a day with every major meal that you eat. Okay. And so this relationship between gastrin and these mass movements of the colon, this is called the gastrocolic reflex. Okay. It's the reflex that triggers a mass movement and potentially defecation when food enters the stomach. okay, or when food is expected to enter the stomach. And this just ensures that the GI tract keeps moving along, right? We've got new food coming, we need to get the old stuff out. So that brings us to the defecation reflex, right? Because defecation is a reflexive activity. It is something that is triggered involuntarily, but thanks to those wonderful skeletal muscle, that skeletal muscle of the external anal sphincter, we are able to time. We are very lucky to be able to time when we choose to defecate, but the actual urge to defecate is a reflex, and that reflex starts with the distension of the rectum. So if you have one of these mass movements and it is enough to drive feces into the rectum, it's going to cause distension of the rectum, the stretch on the rectal wall. When that rectum stretches, those stretch receptors cause a reflexive relaxation of your internal anal sphincter. That's the anal sphincter that is composed of smooth muscle. When the internal anal sphincter relaxes, what you're going to have is that feces that has just filled the rectum is going to move down, okay? And it's going to put pressure on the external anal sphincter. And that stretching of the rectum, that moving, that relaxing of the anal sphincter, that gives you the sensation that you need to defecate. That's when you get that sensation that you need to defecate. And so you're cued to the fact that you have the desire to defecate. And so now it's all about you finding an opportunity to get to a place where you can relax your external anal sphincter and actually have that bowel movement. Now, it's definitely possible to ignore the urge. So if we ignore the urge and we do not defecate, what's going to happen is eventually the internal anal sphincter will contract again. Okay? It was relaxed, but eventually it will contract again, and that feces will move back up into the rectum and maybe even back up into the sigmoid colon. Now, that's fine, right? You'll get another opportunity another time. However, delaying that needs to defecate can potentially cause problems, because that the longer that feces sits in the rectum, in the sigmoid colon, the more water is absorbed and the harder that stool becomes. And so it could put you in a position where you become constipated because you've absorbed too much water from that feces. And now that feces is sort of hard, it's not as soft, and it's more difficult to pass. Okay? And so that does happen to people sometimes. It happens especially to children. Sometimes children will already be constipated, and so when they defecate, it's painful because their stool is hard, and so they don't like that sensation, and so they withhold, they ignore the sensation to defecate, and it just ends up making their constipation worse. So that it's definitely the healthiest to not ignore that need to defecate and to really attend to it when it occurs. So, before we end here, it's worth it to talk about fiber, because we hear a lot about fiber and how fiber is good for us, right? So what's so great about fiber? So first of all, let's define what fiber is. Fiber is any ingested material that is not, any food that we eat that is not digestible by our body. Okay? So when we consume food that we cannot digest, that food remains in our small intestine and it moves on to the large intestine. And what that does is increase the osmolarity of the feces that are in your large intestine. And fiber actually comes in two different kinds. We've got water-soluble fiber, and then we've got water-insoluble fiber. Water-soluble fiber is the kind of fiber that when it mixes with water, it dissolves and it kind of forms almost like a gel. Okay? Water-soluble fiber, an example of this is oat bran. Oat bran is a really good example. Metamucil, if you've ever purchased or used Metamucil, or psyllium husk. Those are, chia seeds are also an example of water-soluble fiber. When you add that stuff to water, it dissolves and it forms a kind of gel. Water-soluble fiber helps to add to the bulk of your feces. And by adding bulk, by literally adding size to your feces, it increases the frequency of you clearing your GI tract, of you clearing your colon. So by adding to the bulk of your feces, every time you have, or close to every time you have one of those mass movements when you eat a meal, it's going to increase the likelihood that that actually clears out your colon. So water-soluble fiber is really good. And we also learned back in last semester's class on cholesterol that when we have a good amount of water-soluble fiber, it also tends to trap bile and cause you to excrete more bile, which means that your liver has to make more, which means your liver takes out cholesterol from your bloodstream. So water-soluble fiber also can help regulate your cholesterol levels. Water-insoluble fiber does not dissolve in water. And water-insoluble fiber is the kind of fiber that tends to have the beneficial effect of increasing the osmolarity of your feces and holding on and trapping water in your feces. And so water-insoluble fiber tends to soften feces. It tends to make it less hard, which is also great because that means that it is not effortful to defecate. So if you've got good amounts of water-soluble fiber, you're going to defecate more frequently. If you've got good amounts of water-insoluble fiber, that feces is going to be nice and soft, so it's easy to defecate more frequently. But there is such a thing as too much of a good thing, because especially when it comes to water-insoluble fiber, if you consume too much of that, it can cause osmotic diarrhea. So that too much water is added to that feces, and it causes the feces to become actually watery, which is not good because that can cause dehydration, in addition to just being very uncomfortable. But that's the story about fiber.